Thank you very much, Jacob, for your introduction. And also thanks for the opportunity to talk about the evaluation of CAR expression and CAR-T persistence, and also our commitment to providing the excellence to CAR-T cell therapy. First, I'm going to share some published data from some literature, and then compare the methods for detection of the CAR-T cell expression, and then share some case studies, and then to introduce ACROBiosystems reagents to support CAR-T cell therapy research at the last. So this is the data from the publication Later Review Drug Discovery. The data was up to March 2020. From this chart, you can see that the global cancer cell therapy pipelines includes about 1,500 active agents, and about 472 more than the previous year's 2019. And the CAR-T cells still occupies the largest portion of the active agents, and also has the largest increase compared to the previous years. And this is the top 10 targets of cell therapies for blood cancer and solid tumors. From the charts, you can see that the top 10 targets for the blood cancer and the solid tumors are pretty different. And so for the blood cancers, you can see that the CAR-T targets still the top one CAR targets, and then BCMA and the others. For the solid tumors, you can see that the tumor-associated antigens occupies is the top targets, and then the HER2 and the others. This is the landscape of cell therapy, and based on this pie chart, autologous CAR-T still occupies the large portion, even though the allogeneic CAR-T has grown a lot. And the current marketed cell therapies are still 5. There are 4 anti-CD19 and 1 anti-BCMA. And there are a lot of active agents in the clinical trials, and also preclinical studies. This is a pie chart of the innovation. So this pie summarized the innovation orientation and the goals for the transforming CAR-T cell engineering. It includes a lot of efforts here to improve either the endodomain part, from endodomain part to the transmembrane and also exodomain, and try to improve the either the CAR-T cells functionality, or they explore more CAR targets to cover more tumors, and to produce faster and safer CAR-T active agents, or overcome the tumor microenvironment challenges, all these kind of efforts. So this slide summarized all the 4 different generations of CAR constructs. The first generation has all the basic elements, including the SCF-way, and then the transmembrane domains and intracellular domains, including the CD3 theta domains, which contains 3 items and which can activate the T cells through the pathway. And the second generation added one more co-stimulatory elements on top of that. And the third generation includes two co-stimulatory elements. And the fourth generation CAR-T also has added IL-12 expression cassette, which can stimulate the innate immune system in order to avoid the immune escape because of the reduction of tumor-associated antigens. And the fifth generation included one more element, that's JAK and STAT binding domain in this construct, which can further stimulate the T cells IL-2 production and activate T cells, which can help to even further prolong the CAR-T cells persistence. And since the method for detecting and monitoring of CAR-T cells is very important in the CAR-T discovery and development, as well as the clinical studies, which can help to understand the underlying mechanisms for the efficacy and safety outcomes. So here, we usually have different levels detection of CAR expression. This is from a literature. Basically, it includes the genomic level detection, transcriptomic levels detection, and proteomic level detection, and organismal detection. And it's the genomic level test usually can use the qPCR and digital PCR to test the CAR vector copy numbers and do the analysis of the integration site. And MR level, which can use RNA-seq message to measure their CAR MR abundance, and with the CAR scope in situ hybridization to determine the presence and subcellular localization of the MR molecules. The proteomic level test is the most practical and efficient method to detect the CAR-T cells or CAR cell surface expression with flow cytometry. This method can also measure the CAR positive percentage compared to the other methods. And at the organismal level, we can do, for example, like BRI and PET scanning to determine the biodistribution of CAR-T cells between or among the organs, or the monitoring the CAR-T cells in the tissue for trafficking at single cell level, cellular level, with the 2-photon microscopy. And these are the tools for CAR detection at proteomic level. Basically, the reagents included the CAR target antigens, anti-idiotype antibody, or the protein L and anti-5 antibody. So the first two reagents, the CAR specific targets, antigen, and the anti-idiotype antibody, can provide one-step staining and also high specificity and sensitivity, and as well can providing a good compatibility with other antibody panels for co-staining of the other interest targets or other indicators. And also the CAR-T target antigens is pretty accessible to all the labs. And anti-typical antibody usually is hard for all the academic labs to access, because we need to develop the anti-typical antibody. Comparatively, the protein L and anti-5 antibodies have lower specificity, but it's easier to access for the lab. And this is the in-house data we generated to compare of the different reagents for CAR detection. And we used the PBMC untransduced cells as the test cells, and without staining as negative control, or stained with phase-labeled anti-5. Sorry, that means I'm really have difficulty here to see the slides even. And also the phase-labeled protein L, and also phase-labeled CD19. So you can see that the phase-labeled CD19 produce the lowest non-specific binding. And this is another set of data generated in-house. We compared the different phase-labeled antigens. And you can see from the left-hand panel, the phase-labeled CD19 FC produced the highest sensitivity compared to the others due to the higher avidity binding activity. And then the right-hand side panel shows the comparison between the phase-labeled CD19 antigen and also the with the phase-labeled anti-FMC63 antibody. And comparatively, the phase-labeled antibody produced a higher sensitivity compared to the antigens. And these are the products we provide to support the CAR-T research. And these are 3 different types of CAR-T target antigen. The first group is fluorescent labeled. It can be directly used for staining. The second group is biotinylated protein products, can be used to staining the CAR expression as well. And the last group is unconjugated proteins, which can be which has the epitope tag, so it can be also used to stain the CAR expression in the flow cytometry. And so I'm going to share several cases which are published by our customer using our company's products, or some data shared from our customers using our products. This is a case published. Basically, this study is about the study the allogenic gene-edited CAR-T cells. The study is transfected with the TCR MR into the activated T cells and transduced with the CAR expression cassette, and to study if the CAR expression cassettes included into TRCA track locus. And this study used the CD3 surface expression as the functional marker to tell if the TCR expression is disrupted. And from this data, you can see that the CD19 CAR expression was detected by using bioattenuated CD19 FC reagent from ACROBiosystems. And it shows that a high proportion of the anti-CD19 CAR T cells existed in the CD3 selective population after the enrichment. And also that's demonstrate that most of the CAR did integrated at the track locus. And this is another case also used by our customer to detect the anti-BCMA CAR expressed in the transduced human T cells. From this data, you can also see that the reagents from ACROBiosystems can differentiate the positive CAR-T rates among different donor T cells. And this is another data shared also from a customer. They used our reagents to detect the anti-CD22 CAR expression in the on the T cells transduced with the CAR. And you can see that the 63% positive rates can be reached. And this is the last case study which detected the customer's own developed BCMA CAR, and they used our reagents to detect their mark CAR-T cells and the BCMA CAR-T cells, and they detected 32% positive rate for their studies. And we have products to support CAR-T development at all stages, including the antibody discovery or T cell activation with all the cytochem products we have, and also expansion of CAR-T cells, QC controls, and also the preclinical study and the clinical studies for the PK study or the immunogenicity study. And these are the hot cells products for CAR detections based on our marketing data analysis. We have all this data, the products available online. And these are the hot cell products. And these are the hot cell products for the CAR detection from the anti-EGO type antibody. And we have strict in-house QC of all CAR detection reagents. This is an example for the PE-labeled anti-FMC63 antibody. We usually have the QC test for the sensitivity, specificity, and also the product stability and batch-to-batch consistency. And this is the last slide. I'm going to finish up with this slide to show that our CAR detection reagents provide excellent performance to all the CAR-T cell research at different stages. Thank you very much for your attention. I'm happy to take any questions. Sorry.
So, Teng, thank you for the presentation. And again, sorry for the technical issues there, but I think good job sticking with it for sure. So we do have a couple of questions, and I think your talk is quite technical, and it brings a lot of provocative topics forward, and obviously keeping track of the pharmacokinetics of these CAR T cells is really, really important to our clinical programs. But a question that's come up is, among those detection methods, which one can provide the most efficient and relevant information on CAR expression? So you've shown us several, but which one do you think is going to be the most efficient and relevant to CAR expression?
Yeah, of course, there are so many different methods to detect the CAR activities and CAR expressions at different levels. But I think the most efficient and relevant methods, probably especially for the clinical studies, are the protein level test with the specific CAR and target antigen or anti-idiopathic antibody in the flow cytometry. The others can provide the methods for different levels and profound mechanism tests, but maybe not all of them are accessible to all the labs, especially for the clinical affiliated labs.
Very good. So thank you for that. Maybe one more question. Among all the different proteomic detection reagents for flow cytometry, which type of the reagent provides the best result? Any thoughts on that?
Yeah, if we're talking about the flow cytometry, usually the specific target antigen can be a very good detection reagents for the CAR cell surface expression. And also comparatively, you can see we have in-house data to show that anti-idiopathic antibody also can be very good detection reagents. But the problem is we cannot, provide the anti-idiopathic antibody to all, different CARs. That's pretty much dependent on if we have the published CAR SFW information to generate the anti-idiopathic antibody. So usually it's not very accessible. So comparatively, I think specific CAR target antigen is still the practical and also specific reagents for most of the labs.
Well, excellent. Thank you so much, Teng, for your talk and for your responses to the questions as well. Very much appreciated. So thank you.
Thank you very much.